I'm going to talk a little bit about modern subtyping, molecules, pathology, genes. I never thought I'd say this, but I'm going to say a little bit about the science of lifestyle influence on frontotemporal dementia. Caitlin Casaletto at our place has really been doing pioneering work, mostly interacting with David Bennett at Rush, thinking about how lifestyle may influence biology. And she's got some nice findings in frontotemporal dementia. I think when I think of FTD from the very beginning time, I started studying this at UCLA, I realized what an extraordinary burden for caregivers this disease was. And in my first cohorts, I had more people die in the caregiving side of this than the actual patients in the first 5 years. So I began to get a sense of how profoundly a disease, as I'll say in a minute, this happens driven by an anatomy in the brain of the patient, which in turn affects the caregivers, how important this dyad interaction is. I'm going to just touch on the BV-FTD phenotype. I never see a patient when I'm not interested in the uniqueness of how this human being with the human condition and the life factors that they have interacts with a degenerative disease in the social brain. I'll touch a little bit on empathy and emotion. Then I'll talk about some of the emerging treatments. It's really been an exceptionally exciting story in the past few years. And I think it's a model for how philanthropy and academia can interact together. So this is a slide I often show, which emphasizes, I think I give Stan Prusner a lot of credit for this concepts in this slide, but the idea is fairly straightforward, that all degenerative diseases have genetic and sporadic forms. I think more and more we're starting to understand what sporadic and genetic means. We've been able to take the genetic forms in particular, model them in cell culture, whether it's iPSC-derived neurons. Amy Gao at our site does beautiful work with worms. Gray Davis has done incredible work with flies, thinking about the FTD story associated with VCP, other animal models as well. So for all degenerative diseases, we have 3 stages. Preclinical, that means there haven't been enough neurons that are dysfunctional to cause a clinical syndrome. Early symptomatic, well, it's different for every degenerative disease. If you have a disease like Parkinson's that begins in the brainstem, don't be surprised if the first symptoms are mood, sleep, and anxiety. That's where this reticular core courses through. If it's FTD, again, it's all social, cognitive, behavioral. And then finally, a symptomatic phase when there is enough brain dysfunction so that person is losing their functional abilities. The Prusner model suggests, I still think it's the best model we have, that these diseases are all diseases of abnormal protein aggregation. Proteins, whether you believe in the prion hypothesis or not, you don't have to believe that proteins in neurodegenerative disease spread from one cell to the next, eventually affecting specific circuits. I think you all know the different proteins we associated with these different neurodegenerative conditions. The frontotemporal dementias, I emphasize FTD, ALS, progressive supranuclear palsy, cortical basal degeneration. These are all FTD spectrum diseases associated with either tau or TDP43 abnormalities. Here's a classic PIC body within a neuron. This is classical Pick's disease, a type of frontotemporal dementia. Here, and this is the protein that links ALS and FTD. You see within a nucleus, the TDP43 protein has left the nucleus. It's aggregating in the cytoplasm of this large neuron, causing dysfunction of protein synthesis and neurodegeneration. I think that increasingly, and Adam Boxer at our site thinks about these diseases this way, that maybe we can treat all the tau forms of FTD in basket trials. They have different clinical syndromes, different types of tau. But if you have an antibody to get rid of tau in the brain, it might work for all of the tauopathies. Similarly, we're thinking about the TDP-43 diseases in a similar vein. And I think the kind of work that Vivek has done, thinking about what are the genes that are, sorry, this is a family cell phone here. Yeah, okay. So thinking about the genes that are affected gets us thinking about where we can intervene in these genetic pathways. This is, I think, the protein spread story. And this is a nice paper Bill Seeley wrote in 2009, but it emphasizes with each of these degenerative diseases, there is a specific topography. Alzheimer's disease, it is the hippocampus, the default mode network that is selectively affected. In BVFTD, this is the territory of BVFTD as the anterior insula, orbital frontal cortex, thalamo, basal ganglia, frontal circuits. Somatic variant hits the anterior temporal pole and the progressive non-fluent aphasia, a very localized disease for many years in the left inferior frontal gyrus, often begins in Broca's region. So different topographic atrophy patterns. And I think what this paper by Bill showed, looking at the resting bold signal in healthy young people, was that these diseases attack normal functional circuit networks with different functions in healthy controls and spread along these functional connectivity networks in a very specific and predictable pattern. So that is the protein spread idea of the frontotemporal dementias, which I'll come back to. So I was taught, and this was a famous phrase in the era of the 1980s and 90s, don't pick Pick's disease. The idea was it's so rare, you'll never see a case. The second idea was that you couldn't differentiate it from other degenerative diseases. And the lore during that time was all dementia was Alzheimer's disease. And I think the third part was the neurologists who coined this term were sort of saying, you know, this is not where neurologists belong. You should not be thinking about behavior or cognition. And of course, I think the deficit on the other side of this coin, the psychiatrists were saying, well, you know, we don't think about frontal anatomy. This is not, you know, our domain. We're in the world of dreams and psychoanalysis. So I think what happened was that this was really vastly neglected. Then I think really beginning with the discovery by Michael Hutton that a mutation in the tau gene could cause frontotemporal dementia. In the late 1990s, 2000s, people started to realize, you know, Pick's disease. It's common. It's the most common dementia under the age of 60. I don't think our epidemiologic studies come close to the real estimate for the frontotemporal dementias. Almost all ALS patients, I think, have a vulnerability to the frontoinsular neurons. And if they live long enough, would develop FTD. PSP, cortical basal degeneration, are really disorders of frontotemporal subcortical cortical regions. CTE, the two major proteins are tau, TDP-43. Many patients present with a phenotype that is very similar to FTD. So just taking those pieces alone, I think this is a common degenerative disease. But it's also become pretty clear that FTD occurs after the age of 70, and we often don't recognize it. A Dr. So from Korea spent time with us and showed that in our database, about a quarter of FTD was present in people over the age of 65. If you got late onset FTD in this study, you were more likely to have tau than TDP-43 as the underlying molecule. Then I think there's a lot of work that shows that, and Claudia's program is showing this as well, that if you're over the age of 80 and you have Alzheimer's disease, you also have pathology that some of us believe is linked to frontotemporal dementia, including probably about 40% of people over the age of 80 have TDP-43 pathology. There's kind of a raging debate about what that TDP-43 pathology is in the old and very old. I would say Keith Joseph's paper by Busiek in 2022 discusses this. I think that this is TDP-43 is a late form of frontotemporal dementia. So wouldn't that be interesting? I'm not convinced this is true, but wouldn't that be interesting that if you live to over the age of 80, not only are you predisposed to Alzheimer's disease, but you're also predisposed to FTD, and maybe they are linked together in some way. It's a very fascinating debate. Okay, so this is why I went into neurology, this amazing correlation between anatomy and behavior. And what I never suspected when we started FTD research was that it might also tell us something about the genes and the molecules responsible for selective vulnerability. The hardest one in terms of anatomy to molecule is BVFTD. This is, as you can see, anterior cingulate, insular, some degree temporal lobe. Don't forget the basal ganglia atrophy and also the thalamic atrophy associated with these conditions. In our cohort, about 2/3 of our FTD patients have TDP-43 if they have behavioral variant. But as you get older, tau becomes more common. Many of these cases are genetic. This is a group of patients who sometimes have the C9orf72 mutation that causes ALS. So BVFTD, most common early age of onset form of frontotemporal dementia, strongly genetic, tilted a little bit for TDP-43. Somatic variant, this is what Pick originally described, focal anterior temporal lobe atrophy, rarely genetic, even more remarkable. When it is genetic, it's almost always, if you have a semantic presentation, a tau mutation or a TDP-43 mutation. 85%-ish of these patients have TDP type C as the molecular aggregate. And then finally, the progressive non-fluent aphasia. In our cohort, 85% of these patients have tau, 50% have cortical basal degeneration pathologically. Most of the other patients have TDP-A associated with a progranulin mutation. So I think in this subtype, we often know if we do measure for genes and the genes are negative, that this is going to be a primary tauopathy and that we can treat these patients in the same way Claire Hinchcliffe thinks about treating PSP as a primary tauopathy. Here are the molecules shown, just very briefly. We have the tauopathies, the first three squares, classic Pick bodies, 3R tau. This is the astrocyte, thorny astrocyte of PSP, lots of aggregate of 4R tau. Here is the astrocytic plaque of cortical basal degeneration, 4R tau. Here are the three TDP subtypes, A associated with progranulin, B associated with motor neuron disease, and C9orf72, 73. and then TDP-C, these aggregates of TDP and neurites. Just a couple comments. I'm not talking about the FTLD fuss, but it's a fascinating story, almost always sporadic, causes a BVFTD syndrome in very young people. I mean, people in their 20s and 30s, high prevalence of psychosis. So I believe many of these people are never diagnosed as neurodegenerative. They are diagnosed with schizophrenia and die with that diagnosis. Here's CTE, here's the tauopathy part of this story. Very important work done by Ann McKee and NFL players. I don't think we know yet how many people who suffer head trauma not playing football have components of chronic traumatic encephalopathy, but a wicked tauopathy that seems to begin in areas around blood vessels, areas of the brain that are susceptible to trauma, and spread out along specific brain circuits. Just a comment, this is a frontoinsular neurodegenerative disease. And I think to understand FTD, we have to understand what these frontoinsular brain regions do. I think the insula may be the most important brain area that we have as human beings, and one of the least studied. So I think this is a particular fascination for me, which I'll touch on. This is a very interesting part of the FTD story that Bill Seeley has developed. The idea is that we have these very large layer 5 neurons called von Economo neurons. They're even larger than adjacent pyramidal neurons. What happens probably in the earliest stage, Bill Seeley calls this stage 0 of FTD, is the nice aggregated TDP-43, which sits in the nucleus. And as Don Cleveland says, regulates the expression of about 110 proteins that either are up or down regulated when TDP-43 leaves the nucleus. So there is loss of this RNA regulation that is such an important part of frontotemporal dementia due to TDP type B. So this is the image. Adjacent pyramidal neurons are well preserved, but it is these phylogenetically new von Economo neurons that we don't understand very well that sit at the site of the beginnings of neurodegeneration of FTD. So the fork cells, very phylogenetically similar, and the von Economo neurons are selectively vulnerable. And the earlier you get in the process of frontotemporal dementia, the more selective the injury is. So this is a nice study where Bill and his group looked at VENs, fork cells, and adjacent pyramidal neurons, and showed that the earlier you were in the FTD process, the more likely you were to have selective vulnerability of von Economo neurons and fork cells. So you can see early stage 0 von Economo neurons are affected much more than the adjacent neurons. Selective vulnerability in a fascinating brain region in a fascinating type of neuron. Same thing happens in von Economo neurons with the tauopathies. Nice study by Dr. Lin and Nana. And what they basically showed that in these von Economo neurons and BVFTD type syndromes, you saw increased hyperphosphorylated tau inclusions, selective vulnerability of a neuron. Why, how? I think it's one of the big underlying questions of FTD and probably has great relevance to therapy as well. Okay. So 3 main genetic risks, and they've all been discovered at different times. All of these genes and their mutations have changed the way we think about the frontotemporal dementias. The first mutations were found in MAPT gene by Michael Hutton at Mayo Clinic. And the idea is that an abnormal gene on one of the chromosomes, it's almost never homozygote. We've had one homozygote carrier and the disease presented very early, but most of these are heterozygotes. An abnormality in one of these tau genes leads to excessive tau aggregation, neuronal degeneration in frontotemporal circuits. Clearly, the way to treat this, if we can get in early enough, is to turn off this abnormal tau signal or figure out a way of degrading abnormally phosphorylated tau. Those are the two strategies that are moving forward. I'm optimistic that in families with tau mutations, if we get the right intervention, can turn off enough tau, can do it early enough, we will have a cure, the first cure for a type of frontotemporal dementia. Progranulin took a long time, a full 8 years before the second major frontotemporal dementia gene was discovered, a little later age of onset. It's a fascinating disease because it's not always, but usually very asymmetric. If it hits the left side of the brain, you get a progressive aphasia. If you hit the right side of the brain, asymmetrically, you tend to get a behavioral variant syndrome. Progranulin is a haploinsufficiency. If you're haploinsufficient in progranulin, don't produce enough of it, you are not only prone to BVFTD and PPA, but you are also more likely to get Parkinson's and Alzheimer's disease. We recently had a very interesting CPC of a gentleman who had a very rapid Alzheimer's syndrome that was followed clinically by BVFTD. And pathologically, we saw about equal representation of Alzheimer's, which we think caused the early symptoms and progressed rapidly, probably because of this haploinsufficiency. And then on top of that, the BVFTD, which took over in the later stages of the disease. C9orf72, it's the most common mutation in our cohort. MRI tends to be symmetric. Lots of involvement in particular of the occipital lobe in this, which I think makes it very confusing for movement disorder groups. What that means is sometimes you don't see the massive frontotemporal atrophy that you see with other forms of FTD. And I think it can be misleading in terms of understanding the clinical syndrome. It's caused by a massive hexanucleotide repeat. Obvious therapy is to figure out a way of turning off this bad gene. We have Claire Cleland, who works with us at UCSF, who is working on a CRISPR approach. And I think within the next 3 years or so, she will be moving this into clinics. It should work. It should prevent the disease. And wow, what a breakthrough that will be when we have therapies for these relentlessly horrible degenerative diseases. This is a national effort led by Adam Boxer, Brad Beauvais, and Howie Rosen. The key for a disorder like FTD, where we don't really have enough patients for clinical trials, is mass force. Get as many centers as possible involved with the phenotyping of people with FTD, better recognition within our communities, and longitudinal studies, just like the ADRCs, to understand how these diseases progress to help us think about therapies. We had an international conference. This is, I think, the dilemma of 2022, even though we had it in 2012. Lots of mutations in families in Europe, Australia, North America, very few outside of those regions. And the question still remains, are these mutations that are European predominantly? Maybe. This is a really nice slide, which I think shows that if you have C9orf72 shown here in dark blue, you know, you are most likely to see that mutation in Northern Europe. With progranulin, we have regions like the VAS region where a founder effect is present, and many family members have FTD driven by progranulin. Tau mutations, Italy, some in England as well. But I think this is really reflecting probably a founder effect of mutations, a single ancestor who spreads this disease across a region. So that brings up the question, is there really no FTD in China, Korea, South America? Well, I think we're finding out that that's just ascertainment. And so this is one study that I've helped to lead, which is a South American initiative where we're hoping to study 6,000 people across Central and South America. A third of them will have Alzheimer's, a third frontotemporal dementia, and a third will be healthy controls. And we're already seeing in these studies being done in places like Peru, Brazil, that we do have genetic FTD. We do have familial forms of Alzheimer's disease. I think one thing I'm really excited about this is we're starting to see about 30% of the people that we have collected so far have Indo-American ancestry. And so I think this will be a deep dive in thinking about whether African populations across the Bering Strait 18,000 years ago came to North and South America, have forms of FTD. It's been a really, despite COVID, an incredibly exciting project, maybe the most exciting one I've ever been involved with. Okay, so going back to this preclinical, there's a long clinical status without too much change. Then just like Alzheimer's disease, maybe 10 years before the onset of the syndrome, we start to see atrophy. I will say each of the genetic mutations are slightly different. When people start to become symptomatic, we see massive atrophy going on, marked increase in biomarkers of axonal injury, and usually a pretty rapid progression to symptomatic. At least with some of the genes, we get a really marked inflammatory phenotype as well as this disease moves forward clinically. This is work by Susie Lee, and this captures the heterogeneity of the progranulin mutations, different than C9 and MAPT, I think, in the heterogeneity. But what she points out is years, but not many years before people become symptomatic, they start to show hyperconnectivity in the network that will eventually lead to hypoconnectivity and clinical expression. So people with non-fluent aphasia, a little bit before they develop the non-fluent aphasia, begin to show hyperconnectivity in Broca's region in this left fronto-insular language network. If it's a BVFTD, it's in the salience network. If it's cortical basal syndrome, it's in this dorsal cortical basal network. And for those people with progranulin who are getting Alzheimer's disease first, it is in the default mode network. We're hoping that studies like this will allow us to know when to intervene and maybe prevent the change of hyperconnectivity to hypoconnectivity and clinical loss. So I think this is a very interesting imaging story. The other thing I would emphasize about clinical trials for BVFTD is that it goes rapidly once someone starts to deteriorate. In this study by Sharon Shaw now at Stanford, Sharon showed that the atrophy in this small study could be picked up in the progranulin patients who had become symptomatic within 8 weeks. So it's a malignant, rapid course in FTD once the disease starts. And in some ways, it gives us an advantage in terms of thinking about whether we can detect the effect of a biomarker. Neurofilament looks like it's going to be a good one for FTD and ALS. This is a nice work by Julio Rojas, a tremendous neurologist, scientist. And he's showing that people with dementia due to BVFTD show a very high plasma neurofilament. When they're in the mild, early symptomatic stage, it's starting to rise, and it's perfectly normal in carriers who are not developing the clinical syndrome. It's a little better for progranulin and C9orf72, but I think as Adam and his groups approach the FDA, they are really going to emphasize this as a potential biomarker to show the efficacy of a neurointervention. Lifestyle, just a word or two about this. It's a small study, but it's done by Caitlin Casaletto. And basically what she showed is she looked longitudinally at the all FTD cohort and looked at the mutation carriers for C9orf, granulin, and MAPT, and self-reported activities related to cognition and physical activity were correlated with slower progression. That's shown here in this slide. So if you had a high baseline cognitive activity, you had slower progression. Similarly, if you had high baseline physical activity, you progressed more slowly. Is this definitive? No. Is it the first effort to begin to understand lifestyle and FTD? It is. And I would say the data is becoming very compelling around Alzheimer's disease. Many different ways of looking at that. I'll just talk about UCSF work, which makes me think that Caitlin's study was plausible. She's had two really nice publications this year, and both of it which suggests that physical activity is associated with better cognitive aging, reduced evidence of neurodegenerative disease biomarkers. One of the things that she looked at with David Bennett was the activated microglia, and people who on actigraphy showed more physical activity had fewer activated microglia, a less pro-inflammatory microglial state, and better synaptic biomarkers as well in this Alzheimer's dementia study. So I think even in FTD, genetically programmed, even close to the age of onset, it may be that these interventions are low-hanging fruit that we should really be thinking about. Okay, here's the FTD syndrome. It really shows you why neurology and psychiatry have to work together. I mean, these are all psychiatric symptoms. Most of the patients, I would say, have at least 4, if not 5 of these criteria. I'll also emphasize that only one of these criteria is a traditional neuropsychological measure, executive loss with relative sparing of visuospatial function. So if your neuropsychologist writes, this individual had executive loss, and this is probably FTD, be wary. They need the behavioral syndrome as well. I think you can have Alzheimer's patients with an executive pattern, lots of Parkinson's patients with an executive pattern, but we need the behavioral syndrome, the core syndrome, to really make a diagnosis of FTD. We developed these criteria in 2011, and they're still used in clinical trials. At least 3 of the 6 possible. You need atrophy on MRI to meet probable, and definite if you have a known mutation. All right, so what do we see? We see lots of changes in reward-based behavior. This is Roll's writing of the orbital frontal cortex. There's medial orbital frontal cortex, lots of connections to the hypothalamus, primary reward-related drives like eating. Lateral orbital frontal cortex, very strongly connected to the insula, the amygdala, all sorts of warnings, punishment, sorts of signaling. Beware, doing too much eating, too much shopping, you name it. That's the lateral aspects of orbital frontal cortex. This area is stage 0, again, BVFTD. This is where BVFTD begins in von Economo neurons. And so no surprise, we see lots of reward-related changing. Alyssa Rezende from Brazil wrote a beautiful paper recently, which showed that about 7.5% of our BVFTD patients start with alcohol abuse as a prodromal feature. In Alzheimer's, the number was about 2%. So I think this just touches on the reward-based behaviors that begin to become unraveled associated with BVFTD due to the orbital frontal cortex injury. Disinhibition, again, with the orbital frontal injury, we've seen crime associated with BVFTD, almost 40% of the patients. So I think when you see an elderly person who commits a disinhibited crime, I think you must begin to think about BVFTD. Loss of empathy, I think this is one of the most consistent findings from our research. Again and again, this is Kate Rankin. If you have loss of empathy by however we measure it, you tend to have atrophy in the right anterior temporal lobe, right ventral striatum, right orbital frontal cortex. So degeneration in this social circuit involved with seeing emotions in others, feeling the emotions in others, and doing something altruistic is a core early loss in BVFTD. If you have right temporal lobe degeneration as the first manifestation of BVFTD, at least half of our patients, the relatives say these people showed loss of empathy as the first sign. The other half, it's more cognitive. I don't recognize that familiar face. Just a word about, I should add Virginia Sturm to this, but I think these are people who really pioneered the study of the neuroscience of emotion. Duchenne, electrically stimulated muscles in the face associated with emotion. Paul Ekman codified that system in the face with facial action coding. Bob Levinson and Virginia Sturm now have studied how emotion is dysregulated in our patients with frontotemporal dementia using Paul's facial action coding. So this is Virginia Sturm's greatest hits. I asked her to put this slide together. Loss of embarrassment reactivity when you're asked to sing My Girl in the laboratory. You're surreptitiously recorded, then the tape of you singing My Girl is played back. This is the real karaoke of emotion. What we see is markedly decreased expression of embarrassment, lack of facial action associated with embarrassment, less emotional reactivity as well autonomically, all associated with this effector piece of embarrassment, degeneration of the anterior cingulate. If you are not disgusted by disgusting films, indeed they are disgusting. I have seen some of them. I have very low threshold for disgust. I can't even think about them, more or less describe them, but disgusting tapes, and you don't show that autonomic reactivity. What you tend to show is profound loss of tissue in the anterior insula, anterior temporal lobe, slightly right-sided predominant. And then even primary regulation of parasympathetic and sympathetic activity is found in our patients with prodromal and early FTD, related, I think, to the insular regulation of parasympathetic and sympathetic activity as well. So high heart rate variability, this is a parasympathetic tone. is associated with social engagement, empathy, emotion regulation, positive emotion, pro-social behavior, optimism, and agreeableness. And we think that some of this social dysregulation is related to the inability to feel what we're supposed to feel related to others' conditions. And this is Dalai Lama, be kind whenever possible. It is always possible. I think our leader in Russia probably didn't train under the Dalai Lama. I'll put it that way. Okay, so marital dissolution, infidelity, no surprise, greater in BVFTD than any other dementia. Empathy loss was associated with a much greater likelihood of marital dissolution, and BVFTD patients who experience marital dissolution or infidelity had lower empathy scores than those who did not. This is an interesting way to look at it. This is Alice Swa. She looked at the patient's brain and then looked at caregiver psychopathology, caregiver global health. Big study. Punchline is caregiver psychopathology and global health was driven in this large cohort by how much atrophy there was in the anterior insula prefrontal cortex. So where the degeneration hits determines how well the caregiver does. I think this helps us a lot think about interventions for caregivers very early. They are very vulnerable to the effects of this illness. I think I'll take about 4 or 5 minutes to end and just talk a little bit about some of the basic biology. So this is a project started by a family where a progranulin gene had wreaked havoc on the family. And they asked to find a cure for progranulin deficiency. Different people have played a major role in this. Bob Faris has developed progranulin knockout and knock-in mice. Eric Roberson, Lee Gann have studied behavior in these mice. Amy Gao has thought a lot about granulin pathways and progranulin pathways and inflammation. We've done high throughput screening. Eric Roberson has been working on transplanting progranulin into mice, I think with some very interesting effects. We have a clinical component, iPSC-derived neuron component, increasingly interested in the lysosome. And there are now, I think, 7 companies interested in delivering progranulin to the brain of gene carriers. We don't have enough patients to really satisfy these different studies. It's really why the studies in South America, other places, are so important. There are 2 potential mechanisms. One is loss of progranulin. The other is increases of different spatial conformations of a granulin unit. There are these different granulin units all involved with inflammation that are cleaved off the large progranulin molecule. Amy Gao wrote this nice Nature review paper that shows that when microglia are granulin deficient, they release pro-inflammatory cytokines. There's a large involvement of C1q, complement protein complex, and this leads to activated microglia, but also deficits in the lysosome. So these are the pathways, the cascades that are released when someone in their brain and in their body does not produce enough progranulin. Nice work here done by Lee Gann, Gretchen Krabbe, where they showed that progranulin deficient microglia release TNF alpha. This in turn has effects on A beta aggregation. And so this may, to some extent, explain why we also have increased susceptibility to Alzheimer's disease with progranulin deficiency. David Perry from our group quantified the granulin mutation carriers that we had and basically showed that we saw very similar OCD-like behaviors in the mice as we did in our humans with this deficiency. simple stereotypies, repetitive checking, repetitive grooming, collecting. So I think the mouse model that Lee had developed had many features of the human condition. Eric Wong has done a series of really brilliant scientific work around progranulin deficiency. And he's basically shown that you can restore thalamic microcircuit function, mitigate OCD-like behaviors, and improve survival in mice who are progranulin deficient if you remove C1qa. So this really points to injury to a specific circuit, complement driven, that in turn drives a very specific behavior, interesting to progranulin, but also interesting, I think, to psychiatry more broadly. This is Dr. Arendt's work with Eric Roberson, but this is AAV approach to rescue cellular deficits in granulin negative mice. And I think the findings have been very positive. And there are a number of companies beginning phase 1A studies, seeing whether we can really change the course of these gene carriers. Just maybe I'll make this my last slide because I think we should have a little time for questions. I think one of the things that we have realized is that if you are homozygote for a progranulin mutation, this was great work done by Sam Berkovici in Australia. He saw brother, sister who were called neuronal ceroid lipofuscinosis. They had retinal disease, cerebellar changes, seizures. And then when they looked at the genes, they were homozygotes for progranulin. So that made our whole field begin to wonder, is the lysosome the critical place where this progranulin deficiency is causing neurodegeneration? Lots of research going on in this space. So here's the paper by Smith et al. In Australia, Almeida in Massachusetts, Portugal. But basically they showed that there is in homozygotes for progranulin, lysosomal storage disease, NCL, heterozygote mutation carriers show NCL-like storage material in the CNS, retina. This is Michael Ward now at NIH's really, really brilliant work that told us that this really does have features of a lysosomal storage disease in addition to the inflammatory story. Jen Yokoyama has found another gene, MFSD8, mostly in underserved minority populations, Latinx, Asian, that homozygosity causes an NCL, heterozygosity predisposes to frontotemporal dementia and maybe Alzheimer's disease as well. This is a big story, I think. Heterozygote disorders cause something different than homozygote disorders. And I think this has lots of overlap with TREM2 in the Alzheimer FTD space as well. Just a very interesting set of studies that bring the lysosome into interest for all of us. Not going to talk about the tauopathies. I think I've probably run out of time. What's happening? I think we're getting really good at diagnosis of FTD. Our hope is that every patient with an FTD syndrome, by biomarkers alone, we will be able to determine what subtype of FTD you have. That'll be really great for clinical trials. New causal risk genes. Even more important, I think, is Vivek Swarov's work, thinking about gene systems that are affected, leading to BVFTD, thinking about where in those systems we might intervene. We need more patients, so big studies, also now thinking about Africa. Tau lowering, Claire Clellan is working on CRISPR for tau mutations, antisense approaches by Tim Miller at WashU. For TDP subtypes, Peter Ubankov has just been approved and just starting an anti-inflammatory compound for somatic variant PPA. Huge number of studies of antibody delivery of progranulin. Denali Therapeutics has a really interesting delivery system via transferrin, genetic silencing approaches. So very exciting time in the future, but also a very exciting time now to help families and patients. Thank you.
Great, thanks a lot. This was an amazing talk, and I think a lot of us have a lot of questions. So let's get started with some of the questions. Please, if you have a question, you can put it in the chat, or you can just unmute and speak up. Okay, while the questions are coming, I have a quick questions. What do you think about studies from our group, from other people's group, suggesting that there are shared changes in all of these neurodegenerative diseases, from Alzheimer's to FTD to other diseases, ALS? And what do you think it represents? Does it only represents the kind of insult and secondary changes that happens when you have an insult to the brain, or does it tell you more about the common actual mechanisms that how brain changes when you have a disease?
Yeah, it's a great question. And, I think Vivek, you are much more able to answer this, but I'll give you I'd love your 2 cents too. It's more, in your case, it's more than 2 cents. It's, it's worth the bank to hear your answer, but I would say both. So I think sometimes we are, seeing the effects, non-specific effects of a systemic brain disease, or even I think in the case of progranulin and maybe C9orf72 as systemic disease, period. In other cases, we see very specific changes that are targetable, and that if we could change, that specific pathway, that we would have a powerful intervention. So, I'd be curious how you would answer this. How do you sort those two out? But to me, this is one of the most exciting, precision medicine efforts going on, and it'll have relevance to every disease that we study.
Sure, yeah, right. I mean, my 2 cents is that we see same changes in glia, in autism and in Alzheimer's disease. So that tells us that there are shared changes that are just reactionary changes, but of course there are the disease-specific changes also. I have one question. Can you elaborate on the caregiver psychopathology?
Yeah. So the burden is profoundly higher, measured by our nurses, caregiver burden in FTD versus Alzheimer's. There are all sorts of sort of paradoxes, which says we better move carefully in terms of how we intervene. Let me give you one example. So our caregivers who seem to be most empathic by a number of our measures are actually the caregivers that are most vulnerable to psychopathology and neuropsychiatric symptomatology. So it isn't as simple as like teaching all caregivers how to be empathic, and what Bob Levinson has worked on is thinking about, a predictor of caregiver vulnerability. There are probably genetic pieces to this, which are similar across Alzheimer's and frontotemporal dementia. There are environmental factors. I mean, I interacted a lot with South America during COVID, and I mean, one of the first things I had to do was just getting our doctors masks down there. I mean, in terms of, and then you think of people in rural Peru who had to work, whose caregiver, had to work, had to keep the patient with FTD in the house wearing a mask. I mean, there are socioeconomic determinants of caregiver health that are just profound. And so I think it's a mix of things that are, really important, genetic, how the disease manifests. But Kate Poseen at our place has developed something called Care Ecosystem, which basically we got a big grant during Obamacare initiative to show that a system would decrease cost for dementia. And our system is a caregiver navigator. So this is someone with a college degree, but not, PhD or master's, who works with a family, is available 24 hours a day to interact with the family, and then back and forth with the care team. And some of the early findings from this data are that you have fewer emergency room visits if you have a care team navigator. I think it's starting to hint at diminished cost. And so I think there are a lot of things, that we should institute with our dementia cohorts that not only are, and all sorts of data on caregiver well-being, patient well-being with this care team navigator. So, I mean, I think these things are cost effective as well. So I think the more that we can initiate these as part of powerful centers like yours and ours, I think the more we can change a little bit the world.
Great. Claudia, you have a question?
That was awesome, Bruce. I really enjoyed the talk. You can't not comment, however, on your thoughts about TDP in late life as a version of frontotemporal dementia.
I'd love your thoughts. I'll tell you the way it's been framed to me. Our pathologists, Leah Grinberg and Bill Seeley, don't think it's a form of frontotemporal dementia. Keith Josephs gave a pretty compelling talk where he said this has all sorts of features of TDPA, and this is really a TDPA related form of frontotemporal dementia. But what are you finding in your group? I mean, I think it's still up for debate, and maybe it's both. Maybe it depends on, we just don't know a lot about TDP-43 yet in elders.
I guess I'm getting so tangled up in terminology and I mean, for example, if somebody has amyloid, it doesn't always have to be a version of Alzheimer's disease. Inclusion body myositis is not considered a version of Alzheimer's. Even if somebody even has tau, like in PE, for example, nobody feels the need to so these proteins, and you're pointing out also, and we're noticing everybody has all these multiple proteins the older they get. But I'm trying to understand better the argument about why, just because TDP is found in these individuals, we should think of it as FTD. I mean, what's the other reasons to think of it as a version of FTD?
Yeah, I think Keith would argue, I'm arguing for him, is that these have morphometric and other features of TDPA that make it seem like it's a form frus of late onset FTD. That is Keith's argument. Yeah. I'm not sure that's going to bear out. And I'm hearing from you, I mean, you may not believe that either, but Keith's a good scientist. I mean, and Dennis Dixon backs this point of view. So I think that's sort of where we are right now with it.
Yeah, no, I believe TDP is related to cognition, just like I also finally believe that amyloid is and tangles are and all that. The problem is it's been 40 years of believing this and I can't tell what to do with it. Can I ask another quick question? This hyperactivity that you see, do you think that that's just a compensatory mechanism in the preclinical stage, or do you think that, because we see that also in other, even in Alzheimer's disease and everything, or do you think that it is something intrinsic to the causal pathway that maybe is a target for intervention?
Yeah, it's such a great question. And let me put it this way. So I've written a little bit about art and the brain and FTD. And so we're now working on the biggest paper on this we can write right now. It's one of our fellows, Deet Friedberg, and she's got 18 FTD artists that have developed FTD in the setting of neurodegeneration. And I think I can't spoil her punchline yet, but I think the theme is increased volume and some connectivity changes in regions that wouldn't surprise you associated with the artists versus the non-artists. One of the coolest findings in this study is that the brain region that is clearly bigger and more connected is the hand piece of the motor strip in the artist. So I interpret this as repeated painting of these people, even in the setting of a neurodegenerative disease. Mind you, a degenerative disease that doesn't affect that circuit particularly allows learning, synaptic pruning, and even increased volume. So, but I think it's also probably, to answer your question, I think it's tantalizing to think that maybe that this hyperconnectivity is a little bit like what Eric Wong has shown in his thalamic microcircuits, that there is an inflammatory change in this circuit that is deleterious and maybe, the beginnings of the neurodegenerative disease process. And I don't know which one it is or which combination. Gosh, the questions you're asking are so central to the understanding of this. I think it's a fascinating question. And Helen Zhu, now in Singapore, showed in 2010, much to our surprise, actually, that just like you said, that our Alzheimer patients actually, compared to healthy controls, had increased functional connectivity in the salience network. So more connectivity in a brain region and less connectivity, obviously, in the default mode network. So I think some of that is just release of one brain region's activity increases activity in another. Yeah.
Wow. Thank you. Thank you.
Yeah.
Great. Thanks a lot. Thanks a lot, Dr. Miller, for joining us. This was an amazing pleasure. I hope you had a good time in UK and you come back here, and please consider visiting us when you are back here.
I can't wait. I just reminded how many friends I have there and how much fun I'd have talking with you all. So thank you.
Thanks a lot.
Come soon. And I want to talk to you about the global work, especially.
Yeah, we'll set up a call. Thanks, Claudia. Okay. Bye all.
Bye-bye.